Hi everyone, I'm Doran. I'm a junior majoring in biology at Stanford University, and I study the ecology of microbial communities in the human gut microbiome. Today I'll be showing you how to make a type of plot that I very commonly make using 16S microbiome data, which is a plot of unique OTU counts, which are basically a crude measure of diversity within a microbial community. Let's take a look at an example of one of these plots here. So in this plot we have multiple panels where each panel represents a set of microbial communities coming from the same source community, either A, B, C, or D. This plot comes from an experiment in which each of these source communities was treated with antibiotics at one of three different doses, either 0 as the control, or 2 or 8 micrograms per milliliter. Each different color of line in each panel represents one of these different doses, and the x-axis here is the passage of the community, which is basically a unit of time, and the y-axis is the count of unique OTUs in that community. OTU stands for Operational Taxonomic Unit, and basically refers to a set of short sequences of DNA from a particular gene, in this case the 16S gene, that have enough similarity to each other that they have been lumped together and estimated to be from one species. Therefore, each unique OTU in our data essentially refers to one species, and so we'll use the terms fairly interchangeably. So looking at the counts of OTUs in each community basically gives us a rough estimate of how the number of species in the community is changing across passages and with each antibiotic dose. So how do we go about making this plot? Let's first talk about the data format that we'll need to use. What I'm showing here is a data frame object in R, which I've imported from a text file. It's in the long data format, which is what we want. What this means is basically that each row is a separate observation, while each column is a unique sample variable that's associated with that observation. In this case, which is a pretty typical case for any type of data that you'd have if you're working with 16S data from the microbiome or any bacterial communities, our data points are basically the number of times that various bacterial species are observed in each unique community. So the number of unique observations that we're going to have is going to be the number of different species of bacteria times the number of different unique communities that we have, and the value for that each observation is going to be the number of times that bacteria was observed in that community. To break this down a little bit further, you can see in our data frame here that our first column is this OTU column. You can see that the observations here are all these sequences of A's, C's, T's, and G's, so they're DNA sequences. The different communities that we have are defined by these next three columns, this source community, passage, and antibiotic concentration columns. We have four distinct source communities that we can see here, A, B, C, or D. We have three different passages. We have 1, 2, or 7. And we have three different antibiotic concentrations, either 0, 2, or 8. And so 4 times 3 times 3 leads us to have 36 unique communities. And so that means that in order for every observation to be a unique combination of bacterial species and community, we have 36 rows per bacterial OTU. This next column here is the value that's associated with each of those combinations, and so this is the number of times that each OTU is observed in its respective community. And the rest of these columns here are good examples of why long data is useful. These are all taxonomy columns. They're just additional information that we've tacked on to the end of this data frame in the form of additional columns, and they tell us the taxonomy associated with each particular OTU. But they don't increase the number of rows in the data frame, and they're quite easy to add or remove. So this is the advantage of long data, is that you can very easily add on new columns or remove them or do other data manipulations using R packages like dplyr. So how do we go about actually making this kind of plot? Well, in order to do this, we're going to actually need to add a new column. And the column that we're going to need to add is basically one that tells us how many unique OTUs are present for each unique community. So we're going to use the dplyr package to do this, and we're going to use the functions filter, group by, summarize, and n. You can see that this is where we're going to do this data manipulation. So we have this data variable that is our current data frame, and we're going to pass it into this first function filter. And what filter is going to do is it's going to remove all of those observations in our current data frame where the count variable is not greater than 0. It's going to keep only observations where the count is greater than 0. And so that's going to remove all those combinations of OTU and community that are in the data frame because it has to have all those combinations but don't actually have any real information associated with them. The next function we're going to pass it into is group by. This function doesn't really do anything by itself, but it basically tells whatever comes after it to run separately on all the variables that we pass it into it. So here we're passing in source community, passage, and antibiotic concentration. So this is going to create basically 36 unique groups, which each have a unique combination of these three variables, and that way we can count up the OTUs, the number of unique OTUs separately for each of those 36 unique communities. The next function we're going to pass it into is summarize, and so this is what does the brunt of the work. Summarize creates a completely new data frame, and it includes columns in that data frame based on the previous grouping variables and on any new variables that you define. So here we're defining this OTU count variable, which is going to be the number of unique OTUs that are observed in the community. And the value that we're assigning to this OTU count variable is the value that's returned by this n function. And what n does is it grabs the number of observations that are in the current group. And so because each group here is basically each unique community, we're only going to have like at most one copy of every one observation for every OTU in this community, and all the observations where the count of that OTU is actually 0 are gone, and so this n function will actually return the number of unique OTUs that are present in our current community. So the final output of this, of all of these functions is this new data frame called data_OTU counts, and what this looks like is a data frame where there's 4 columns. There's these 3 grouping variables: source, community, passage, and antibiotic concentration, and for each of these unique combinations of grouping variables, we have one value associated with the OTU count, so the number of unique OTUs. You can also see that there are 36 total rows here, so you can just confirm that there's one row for every unique community. One thing that's just important to note is this operator here that you put at the end of each line. This is called the pipe operator, this %% sign, and this is necessary in order to string together separate functions using dplyr. So I'm piping in basically the initial data set, and then I'm piping that to the filter function, then to group by, and then to summarize. And then because there's no pipe operator here at the end of summarize, this is the final data frame that's going to be returned to this variable here and assigned to data_OTU counts. So now that we have our data frame in order, we can go ahead and start plotting it. So in order to plot it, we're going to use the ggplot2 package. And so we're going to pipe in our data frame to this new ggplot function call, and this is going to create a new ggplot canvas, and it's going to allow us to access variables from our data_OTU counts data frame. So the next function we're going to call inside this canvas is geomLine, which creates a line plot, and then inside of geomLine we're going to call this ace function, which stands for aesthetic, and inside aesthetic we can define these things that we want to have, like our x variable, which we want to be the passage, our y variable, which we want to be the OTU count, our group for each of the lines, which we want to be the antibiotic concentration, and our coloring for the lines, which we also want to be based on the antibiotic concentration, since we want those 3 different lines with different colors for different antibiotic concentrations. Note that any variables that you call inside of ACE need to be, or any variables that you enter into ACE have to be exactly the same spelling and capitalization and everything as the ones that are found in that data frame that you pass in. And then also I am just putting these passage and antibiotic concentration variables inside this factor wrapper, because this will turn them from a numeric into a categorical variable, and that will allow me to space things evenly despite the passages being like 1, 2, and 7, for instance. This will help alleviate some of those spacing issues. Also note that you can put these arguments that you've put into ACE in the ggplot call initially as well, so that way they'll just hold for basically every plot that you decide to draw on this same canvas. So then, since we also want to have each panel be its own source community, we're going to call this facet wrap function, and we're going to enter in the argument source community. Just note here that you have to have this tilde in front of it in order for this to work properly. And the rest of the variables here are just minor things that change other aspects of the plot. For instance, we have the x label, the x-axis label, the y-axis label, the plot title, and here we change the legend title and the color mapping on the legend. And so that will result you in this plot that we have here, which is what we started out with. So I hope this was helpful for learning some basics of data manipulation using dplyr and plotting with ggplot2. Now that you know how they work, I would recommend that you try playing around with the code a little bit. There's a second data file in the repository that has basically this same initial data frame that we started with, but it has an extra variable somewhere, like an extra column for the replicates. So there's replicate 1, there's replicate 2, and so there's basically double the number of observations in this data frame. And so I'd recommend that you try making this same plot that we have here, but with an additional set of 3 lines on each panel for the second replicate of every community. Good luck, and have fun with your plotting.